much Academy for inviting me to give a lecture, and thank you very much for this wonderful arrangement here. So today I will be talking about some of our recent work which we have been doing, entitled as Tracing the Evolutionary Threads of Myxobacterial Flagella. So I'm sure we are kind of getting delayed for the tea, but I will try to make it a little bit engaging so that you understand what we are doing here. So my lab usually works on sensing mechanisms. So this is knock, like we try to ask the bacteria that how they are sensing the environment, and that's what this story will go on. And Utkarsha has been leading this work, and she will be graduating this year on in the coming year. So we at IIT Hyderabad has two departments related to biology, biotechnology and biomedical engineering. In biotechnology there are several branches, and we do majorly big data biology and microbiology in our lab. And the main question which we have been trying to understand from our work is, I'm sure many of you are aware of DNA, proteins, and DNA proteins have sequences which can be computationally seen, as you can see here, and then these sequences turns into structures which are actually the functional unit of any particular protein, and these functions, they keep evolving. So we are trying to understand this whole dogma of sequence, structure, function and evolution, and to some extent most of our lab projects are revolving around these four things. The major focus area in my lab is on microbes. We do a lot of work with microbes in between on SARS-CoV-2. Also we did some work. Today my focus will be on the myxobacterial genomics. We also do a little bit of medicinal plant genomics and metagenomics. Again, here also we are staying with microbes only. We are trying to understand how plant and microbes are interacting. We are doing a lot of collaborations, and from last 2 years we are also developing several tools which is actually very fascinating to develop, because you can go in any kind of computational direction. These are some of our tools which we have recently made, and some of them are most of them are under review because we started last year only. In case you are interested, we do a little bit of a very off the research kind of work also. We have created one very fascinating tool, Google Scholar Lens, which if you will run on your Google Scholar profile, it will kind of bifurcate or multifurcate your Google Scholar data into authorships, and also create a new index which we are calling SH index. It's a new version of H index, and again this story will be out soon. I'm sure you will come to know about it. Okay, so coming to the main topic of this evening. So we work with Myxobacteria. These are social microbes. They live in groups. What you are seeing here are fruiting bodies of Myxobacteria, and I have been working on Myxobacteria since my PhD, published so many papers, but still there are so many questions which are pending, and we are still looking into it. They are very unique because of several reasons. First is their unique taxonomy. They are majorly aerobic organisms here, but they evolved from anaerobes. So anaerobes means they don't need oxygen for their respiration, but these organisms have evolved to utilize oxygen. Second very important thing, at least for computational person like me, is they have a very large genome size. Very large genome size in terms of bacteria. A bacteria is supposed to have like 3 to 5 MB genome on average. If you see, this is all the bacteria. Sorry, this is a little bit incoordinated, but yeah, if you see, they are all the bacterial genome size, and Myxobacteria lies on the top of it, which means that they have the largest genome size. Even the largest genome has been sequenced by me during my PhD. So, and because of this, and because they are social organisms, they have a very complex signal transduction system. Means they have a very complex sensing mechanisms which allow them to sense the environment. Now, the third most important thing about them is that obviously they have a very complex physiology. They have a very large genome size, so they encode a lot of proteins, and many of these proteins are involved in their social behavior, their gliding, their fruiting body. They make like so many secondary metabolites which are antibiotics, which I will discuss a little bit. They degrade so many biomolecules, and they are very slow growing. So many of these questions, many of these physiological parameters are very fascinating for a computational person also. So Another very important thing about these organisms, which is less explored as of now, is they make they make the largest number of secondary metabolites per organism. So if you see, this is the distribution of all the secondary metabolites. So secondary metabolites, for those people who don't know, secondary metabolites are actually the antibiotic molecules, which when they are within the genome, we call them secondary metabolite, but when they are expressed and come out of the bacteria or secreted out, they usually are very good antibiotic molecules. And if you see here, these are the Myxobacteria, which you can see here, and they make the largest number of secondary metabolites, at least in the whole bacterial kingdom, on median scale. Even if you see one of the largest secondary metabolite producer is Streptomyces, which has been worked on a lot, as you can see by the dots. They make most of these Myxobacteria, they make more than Streptomyces secondary metabolites. So very fascinating organism. But so in the last several years, while working with these genomes, we have come to several type of conclusions and several type of theories. So means maybe many of you will recognize this organism. Okay, Archaeopteryx. Okay, so it's a organism, Archaeopteryx, which we usually say, and we have found it as only fossils. Okay, that it was kind of a connecting link between reptiles and aves. Aves means birds. So it has features of reptiles and birds. Okay, which is very fascinating. So they are, in evolutionary biology we call them missing links or connecting links. So we have found similar kind of things. If you see, these are the bacteria and these are fungi. Okay, so these Myxobacteria, which we usually work on, they have, obviously they are rod shaped, but they make spores similar to how fungi makes these in sporangials. Okay, so these are sporangials of Myxo, which is almost similar to fungi. So sometimes Myxobacteria are also known as kind of a connecting link between bacteria and fungi, but we are going one step further. So these are all our organisms, if you see. So these are Myxobacteria. Okay, and if you see, most of them are 9 MB, they are aerobic, and they have 70% GC content, and their ancestors, which we are saying earlier, they are delta proteobacteria, which are very low genome size, 5 MB, they are strictly anaerobe, and they have a GC content of 45%. So what we have found in the last several years, that these three organisms are kind of connecting link organism between this and this, because they share many properties with each other. So now if you see, their genome size is almost equal to the ancestor. Some of them, 2 of them at least, they are strictly anaerobe, similar to the delta, and one of them have like almost equal GC percentage as delta proteobacteria, and obviously many of them share the similar properties with Myxobacteria. So based on this, we are trying to convey that these are connecting link organisms, these three organisms, and they can tell us a lot about the evolution of these Myxobacteria in a general perspective. Okay. So as I said that we have been working on several of these things, we have sequenced them, cultured them, sequenced them in our lab from last several years, working on several of these directions. Today I will just talk about signal transduction gliding motility. So just to introduce you quickly to the motility, most of the organisms are, most of the bacteria are motile, many of them are non-motile also, where they take support of other organisms to move, or they take help of the host. Okay. But the moving organisms they usually move, the bacteria, they usually move either with flagella. So as you can see here, the thread-like structure, and that's what we call flagella. And sometimes they move by pili. Pili are a small thread-like organism, but very shorter than flagella. And many organisms do move by focal adhesion complexes, and some of them just slide. Okay. So out of these all 5 kind of motility patterns, Myxobacteria depict gliding and twitching. Okay, so they do either gliding or twitching. So just remember that, which also conveys, means I am just trying to give you some preliminary data, that they don't have flagella. Okay, means Myxobacteria are not known to have flagella. You will ask that why the title has flagella. So we will come to that later. Okay. Now most of these motility, they are supported by sensing. So a bacteria who is living in this environment, it will sense the environment. If there is a chemoattractant, it will move towards it. If there is a repellent, it will move away from it. So they do this using signal transduction system, as you can see here. These are the overall bacterial signal transduction system, which can be like one component, which are transcription regulator, where there is only one protein. In two component system, there are two proteins, sensor kinase, which will sense the signal, as you can see here, and respond via this response regulator to the target gene, activation or repression. Now, this is a complicated two component system where the signal is received by this chemoreceptor protein, which we call MCP protein. It sends the signal from outside, like either by this ligand binding domain, and then it will send it to key A, which is similar to a sensor kinase, and then it will send the signal to key Y, and this key Y will send the signal to all the functions. It can be flagellar motility, type 4 pili motility, or so many functions which bacteria can do using these chemosensory systems. So remember that chemosensory systems are one of the most complex sensing mechanism system, at least in the bacteria. Now, we have developed some methodology to identify these systems, and if you see, this is E. coli. So E. coli has this chemosensory system, key A, key R, MCP, and tar protein, and nearby you can see these are flagellar genes. Okay, so the blue ones are the flagellar genes, and the system is kind of go on. So now if you see, E. coli has one chemosensory system. E. coli is a small genome, 4 MB. Similarly, Bacillus is also 1 MB genome. But if you see Myxococcus xanthus, which is a 9 MB genome, it has 8 chemosensory systems, which is the largest in the bacterial kingdom. And even if you see Pseudomonas or Vibrio, which are almost similar to the E. coli, they have 3 or 4, which is a large number for a small organism. So what I have done earlier, we have identified all these chemosensory system across Myxobacteria, and we have characterized them. We have identified almost there can be 14 type of chemosensory systems within the bacteria, within these organisms, and we have also characterized their function using some in silico analysis, and it has been earlier published. Again, we use a lot of, as a computational biologist, we use a lot of phylogeny. In case phylogeny is a difficult word for you, phylogeny, we try to take like similar type of sequences, try to cluster them, and then demarcate each cluster separately. So it's basically similarity based clustering, and if you see, each cluster is kind of making one clade. So if you see this one is one single cluster, key 3. If you see this one is another cluster, key 1. So based on this phylogeny, we can easily classify these kind of systems. So what happened recently, means a few like last year also, we were exploring about this, and we found that there is, so this is our overall classification of chemosensory systems within Myxobacteria, and when we were looking at it, we found that these two systems, ECSS2 and ECSS3, they are only present in these two organisms, and maybe along with this one organism from here, which is very fascinating because this is only present in AD, Andromyxobacter, which is our one of the connecting link organism, as I mentioned earlier. When we look it into the phylogeny, you will see that these connecting link organisms are making a very separate clade or group as compared to all other Myxobacteria, which means that they have a different kind of features and different kind of physiology, even different kind of genome within them. So when we look into this cluster, we found that ECSS3 and ECSS2 are present together, and upstream of it and downstream of it, we have full-fledged flagellar cluster. Okay, so remember that how genome works, that if there are genes which are making some particular type of protein present in the genome, usually it means that it will, it should work, it should express, and it should give some function. Okay, it is possible that some of the key components might be missing, and in that way it is not functioning as it should. So what we realized that in Andromyxobacter at least, they have full flagellar system. So it was very fascinating that, okay, why these organisms have flagellar system. So we looked into in case, just to quickly give you some heads up, that flagellar system is a very big macro machine. It's made up of like almost 30 to 50 proteins, and they work based on the rotation by these mot A, mot B proteins here. Okay, and these kiwi which I was discussing in the chemosensory system, this kiwi, they will regulate FLIM protein here, which is here FLIM protein, and decide that flagella should rotate counterclockwise or clockwise. Okay, so based on chemoattractant, if there is a chemoattractant, the flagella will do counterclockwise motion, which will move forward. If it's a repellent, it will do clockwise motion, which will do reverse motion. Okay, so we did phylogeny of again all those proteins in all the organisms, and we found that this particular system, which is involved in flagella, it is making one separate cluster, as you can see here. That's what we expected, and as you can see, it is only present in Myxobacteriaceae and Labellithraceae and not anywhere else. And as you can see, all the clusters are very well demarcated, F10 separate, F5 separate, F8 separate. We also looked into flagellar gene orthologs, and we found that only in these two group of organisms we have flagellar genes, and in rest of them the genes are completely absent. So the question is that Myxobacteria don't have flagella, they do gliding motility via pili or focal adhesion molecules. So what is happening in these connecting links, and how these connecting links have these full-fledged system. So we expanded our analysis first to the MAGs. MAGs are metagenomes from very niche areas like very deep sea or untouched habitats, or even like local environment like this. So we found that many of those deep sea ocean MAGs, they also have this particular cluster. As you can see, this is F7 cluster, and now F7 cluster has Myxobacter plus these many different type of MAGs, which means that even in those MAGs this system is present. Okay, we looked into all those MAGs and we found that yes, the chemosensory system proteins are in between and flagellar genes are present on upstream and downstream, suggesting that they have those systems. So we again generated another phylogeny and mapped all these proteins, and if you see, the colored ones are MAGs, and these ones, the groups which I have put rectangles, they are the Myxobacter connecting links, and if you see, they all have flagellar systems. Now another thing is that this system is F7 type, and this F7 system is only present in these rectangles. And when you see the sensing protein or MCP protein, only this particular protein 36H, which does help in flagellar motility, it is also only present in these rectangles, which suggests that these organisms have the sensing protein, they have the flagella, and they have the chemosensory system, and all of these three machines are working together to move via flagella in that particular environment, because at one point of time these ancestors or these connecting links, they must be residing or living in an environment where flagella was required, and now because they are living in soil, flagella is not required. So that's why they have lose these flagella from their system along with chemosensory system. So a very big machine or two very big machines they have lost in their life cycle. So this is our final proposing model, that these are the ancestors which has the flagella and the chemosensory system and the MCP protein, all three. But in due course of time, only the connecting link organisms which we are suggesting, they have these partial, or you can say 90% or 95% of the flagellar genes and chemosensory genes, but rest of the isolates which are very well growing in the environment, soil environment, they have lost these three specific systems. So this is our manuscript under preparation, and we are trying to communicate it soon. So with this, I would like to thank the whole lab who has been working with me, all the people. They are all working on very different aspects of Myxobacteria and some other aspects of Vibrio, SARS-CoV-2 and tool making, and we are supported by Inspire and DST, and yeah, ANRF also now. Yeah, thank you very much. Yes, sir. No, I'm here. So, yeah.
So, nice talk. Any questions?
So these are kind of a connection between unicellular and multicellular stuff. I mean, do they have quorum sensing and things like that? I mean,
Yeah. So very good question, ideally, because means we didn't discuss it, or I even I have hidden that slide of multicellularity. So these organisms, so we are multicellular organism. They depict multicellular behavior. So they are social microbes. So they do like swarming as a group. So if and they are very slow growing organisms. So they just keep swarming like this. So if you see on plate, they will keep growing like this. Even if they have to do predation of other small organisms, they will swarm like this and try to engulf them. So yeah, they depict like these kind of behavior. We are not studying them, at least in this study, but yeah, that's kind of a step forward.
so do they have quorum sensing type of signaling
So,
mechanism? Do
so
they?
yeah, so the quorum sensing comes as a very preliminary kind of sensing, ideally. So they don't have that too many. So the one component system is kind of low dominant, two component system are very high. So their major signaling is via two component system. So if you see some other organism, let's say Vibrio do a lot of quorum sensing along with two component system also.
So when they receive signal, they do it collectively.
Yes,
That's what
they
you
do
are
it
saying.
collectively via two component or chemosensory or these kind of behaviors.
So there's RNA-seq data also for these.
So we are trying to find it. So the problem is like the overall problem why Myxobacteria are not becoming very famous, similar to Streptomyces, is they are very slow growing. Very slow growing means you will not see the growth. You will see growth after 10 days or 7 days or 10 days. So in that time, the contamination and all those things will start jumping up. So the only organism for which we have a lot of data, or for which the molecular genetics is very well adapted, is Myxococcus xanthus. So for that we have a lot of data, RNA-seq also, but at least for these connecting link organism, I think overall there are 5 or 10 papers. So we don't have that much prevalence. Yeah. Yes.
How many genomes do you have in this particular sample?
So right now, so this is a little bit old data. Old data means like 2-month old data. So this has 81 complete genomes, and I think 50 metagenomes. But in the latest version, which my student is working, we have taken the whole data set which is available for metagenome, which is like almost 5,000 metagenomes, and around 81. The genome wise it's same, but metagenomes we have increased.
So, two curious questions.
Yes.
One of the thing is the switch from anaerobic to aerobic. So there would be a big metabolic
Yes.
switch. So did you have a chance to look at the extra part of the genome which does not map to the ancestral genome? Does it actually have those kinds of functions in them?
Yeah.
And where would that genome would have come from? Because it would not be by mutation, so it would be some horizontal
Yes.
gene transfer. So did you get a chance to
So
see where that extra, what is that extra thing contributing?
Yeah. No, that's a very fascinating question, because ideally that is the main question of Utkarsha, which she is working on, but she tumbled on this story, and this question is kind of like getting a little bit lagged. But so what is happening with Myxo? They have only 15% genes coming from ancestor. Rest 85% is coming from different clades. Why they have been accumulating that means we don't have an answer right now. But even if you see, let's say this kind of this flagellar system. So this flagellar system, we are trying to find out that okay, it came from like, let's say some other group of organism. So they didn't have it earlier. Okay, even so it's kind of becoming why they have gained this much genome. That is the broadest question which we have. For that, the main first point is that they have been accumulating it extensively and doing duplication and neo-functionalization. So they have been gaining, duplicating, and making new function for those proteins. Why they are doing it, I also don't know.
Could the alternative be that they really are not the connecting link because they have a large genome from some other and they have actually acquired the other part of the genome from the bacteria? Meaning, if you flip it around, because if the bulk of the genome behavior is different, then could the part which you're dealing with is an acquired part?
Yes. No. That's a good point. So now, if you think of it, only one clade of Myxobacter has a small genome. All others are kind of very big genomes, like ranges from 9 to 16. All the other parts. So it looks, means again, like gene gain and loss is kind of very relative term. Someone can say they are gaining, someone can say they are losing, but it looks like that the others have gained, and that's how they have flourished or they have become like this huge.
Okay. Yeah. There's a question there.
Gene transfer, like it has a very big gene size. So is it like acquired a lot of genes due to horizontal gene transfer or something like
So
that?
when any bacteria acquire gene or multiple genes or big cluster from other organism, that is horizontal gene transfer. So whatever we are saying that 70% or 75% genome is coming from outside, it means it's coming via horizontal gene transfer.
Okay.
Yeah.
So is there any evidence that from which organism it acquired this much of genome or something like that?
So means evidence wise we have the data, not in this at least,
Okay.
but yeah, we have the data. Means phylogeny is a very good indicator in that manner. And
Yes.
some of those phylogenies are very clean phylogeny where you can directly see that we have this organism and the nearest organism group is very far away in the taxonomy. So something like if we say human got something from Neanderthal will be like very close by, but like from fish. Okay, so that kind of far away.
Okay.
Yeah.
Thank you.
Thank you.
So I think we come to the end of the session. Thanks the audience for being there.